All right, welcome to The Explainer. Today we're diving into a pretty radical new idea in medicine. What if we could heal our bodies not with cells, but with the microscopic messages they send out?
I know, it sounds a little like science fiction, doesn't it? But this is the core idea that's driving a complete revolution in regenerative medicine. So forget what you think you know. What if we could just deliver the instructions for healing, all packed up in a tiny bubble?
So to really wrap our heads around that, we've got to start at the source, the body's own master cells.
Okay, so stem cells. We've all heard the term, right? They are the absolute foundation of regenerative medicine. They're these amazing kind of blank slate cells that hold the potential to become, well, pretty much anything.
So what makes them so special? Well, it really boils down to two incredible things they can do. First, they can make endless copies of themselves. And second, they can transform or differentiate into any specialized cell the body needs. We're talking heart cells, brain cells, cartilage, you name it. And what's really cool is that with modern science, we can now create these powerhouses called iPSCs from adult skin cells, which really opens up a whole new world for therapy.
Okay, but this is where the story gets really interesting. The latest science is suggesting that the real magic isn't just in the stem cells themselves, it's in the tiny messages they're constantly sending out.
So say hello to the exosome, and the first thing you need to know is, these aren't cells at all. Think of them more like microscopic delivery packages. One scientist even called them tiny soap bubbles, and I love that. They're packed with vital instructions, proteins, genetic material, that stem cells send out to tell other cells what to do.
You know, Dr. Joy Kong put it perfectly. If the stem cell is the command center where all the intelligence is, then the exosomes, they're the workers out in the field, carrying out the orders to repair and regenerate tissue.
All right, let's really dig into this, because understanding the difference between a stem cell and an exosome, well, it's a total game changer. This is the key to the whole thing. Think about it like this. On one side, you've got the stem cell. That's the factory. It's alive, it's got a nucleus, and it can make copies of itself. Then, on the other side, you have the exosome. That's the message, the package that the factory ships out. It's not alive, it has no nucleus, and it can't replicate on its own.
And the size difference here is, well, it's just mind-boggling. An exosome is about 100 times smaller than the stem cell that created it. To put that in perspective, if a stem cell was the size of a basketball, an exosome would be smaller than a grain of sand.
And because they're so incredibly tiny, you can cram an absolutely insane number of them into a single dose. I mean, check this out. A standard vial for therapy might have 30 million stem cells, but a vial of exosomes, it can hold up to 50 times billion. That is a massive, massive difference.
So you're probably thinking, okay, if these things are so great and you can get so many of them, why aren't they everywhere? And that right there has been the huge bottleneck holding this entire field back. The problem, plain and simple, has been just getting enough of them. For this to work in medicine, you don't need millions or even billions. To treat just one person, you need trillions of pure, high-quality exosomes. It's a staggering number. So yeah, if stem cells are the factories making these things, how in the world do you build enough factories to supply everyone? How do you go from a little lab dish to, an industrial scale that can actually help people? Okay, so now let's get to the good part. The incredible breakthrough in biotechnology that finally started to crack this massive production problem. First things first, you have to know what you're even looking for, right? And here it is. This is what scientists were hunting for under an electron microscope. This single tiny vesicle, just 100 nanometers across, packed with all that healing potential. The real aha moment here was a total shift in thinking. For decades, scientists grew cells on flat petri dishes, 2D. The breakthrough was to start growing them in complex 3D environments that are much more like the human body, using things like scaffolds and these advanced bioreactors. And this shows you the next crucial step. See, it's not just about growing the cells. You have to be able to harvest what they make. These advanced biofactories use these really complex systems to carefully separate and purify those trillions of exosomes, making sure the final product is clean and potent. So, how did they know it actually worked? Well, the proof, as they say, is in the payload. Inside these little purified exosomes, they found a unique genetic signature. Think of it like a barcode of instructions, made of something called microRNAs, all ready to be delivered to other cells. And here's the payoff. This is what it's all for. When they took these mass-produced exosomes and applied them to damaged brain tissue in the lab, bam!
Cell death went down, and healing went up. The factories worked, and the product delivered.
Okay, so they solved a massive production problem. That's huge. But what does this breakthrough actually mean for the future of medicine? What does it mean for you and me?
Well, this basically kicks off the era of what's called cell-free therapy. We're talking about treatments that give you all the healing signals of stem cells without actually using the cells themselves. And the potential applications are just wow. Skin rejuvenation, anti-aging, faster wound healing, and maybe, just maybe, entirely new ways to tackle devastating neurodegenerative diseases.
So really, the big takeaway here is that by mass-producing the message instead of the factory, medicine could get a whole lot safer, easier to scale up, and way more precise. We're basically talking about bottling the body's own repair manual.
And that just leaves us with one final, incredible thought. If we can really mass-produce the body's own healing code, what do we choose to fix first?